A very good day, Doctor. My name is Harishwar Pillai Analaki Sidambaram Pillai, matrix number S66238, and today I would like to share a simple presentation on the application of gene knockdown in aquaculture. Welcome to my presentation, Doctor, and today I will explain gene knockdown and how is it applied in aquaculture. So, what is gene knockdown? So, gene knockdown is a genetic technique that reduces the expression of a gene. In biotechnology, there's gene knockdown and gene knockout. So, what are the differences between both of them? Well, gene knockdown is the reduction of gene expression of a gene of an organism, whereas gene knockout is the complete elimination of genes from an organism. There are three methods which is applied in the gene knockdown process, which is RNA interference, antisense oligonucleotides, and CRISPR interface. For RNA interference, it is the introduction of double-stranded RNA to a degraded mRNA. As for antisense oligonucleotides, it is a short DNA or RNA sequence which binds to the mRNA. As for CRISPR interface, it is a genetic editing tool for knocking out specific genes. In aquaculture, the biotechnology of gene knockdown is used in various fish species. However, the most famous or most common species where gene knockdown is practiced is in the giant freshwater prawn, which is Macrobrachium rosenbergii. So, why is gene knockdown widely practiced in Macrobrachium rosenbergii? Well, this gene knockdown is practiced is because males have a faster growth than females. So, basically, in a nutshell, they would like to increase the population of males rather than females due to its faster growth. Gene knockdown in Macrobrachium rosenbergii is not a new thing. However, with modern techniques and modern technologies, it has made the process easier. So before this, farmers practiced microsurgical androgenic gland ablation, where it had a very low success rate and its duration was quite long, and it was quite difficult to identify neo females, which is an intensive labor. However, with modern technologies such as RNAi gene knockdown, it made the process easier, where it silenced the insulin-like androgenic gland in mature males, which produces the neo-females, which has high success rate and is very less laborious compared to the traditional method. So, how does RNA interference in gene knockdown works? So, in simple terms, how does it work? Well, for the first step, which is the target gene selection. So, IAG was identified as the target RNAi. Why? Because it is important for male sexual differentiation in Macrobrachium rosenbergii. Next is the synthesis of dsRNA. So, synthesis of dsRNA, or also known as double-stranded RNA, to the complementary IAG gene. This specifically binds to the mRNA of the IAG gene. The third step is the introduction of dsRNA. So, the synthesized dsRNA is introduced into the prawn system through injection. Inside the prawn, the dsRNA triggers the RNAi pathway. Next is the dicer enzyme action, where the synthesized dsRNA is processed by the dicer, which is an enzyme which cleaves into the small interference RNA or siRNA fragments. The fifth step is the formation of RISC. The siRNA fragments incorporate into the RNA inducing silencing complex, which is the RISC or RISC. The siRNA guides the RISC or RISC to the complementary mRNA of the IAG gene. Next is the mRNA degradation. So RISC binds to the IAG and to the mRNA, which produces the degradation. This prevents the translation of the IAG protein. The final step is the phenotypic effect. So, the inhibition of IAG products blocks the male sexual differentiation, which produces a male polar. So, gene knockdown brings various impacts in aquaculture. As for the positive impacts, the first is the faster growth. Since Gene knockdown produces a monosex population, and in Macrobrachium rosenbergii, it always tends to be an all-male population, and males are known to have faster growth rates, where an all-male production results in a high growth rate. This creates a uniform and a quick growth, which makes the harvesting process predictable and efficient. Next is its simple management. So, an all-male production makes it easier to conduct a breeding program where it's a bit simpler to select or to do selective breeding. So, the third positive impacts of gene knockdown is high profit returns. As for Macrobrachium rosenbergii, the males are the ones that grow faster compared to the females. Thus, with the monosex population, which is all male production, the farmers can enjoy quick profits due to its high growth, which can be produced in a shorter time. Next is the cost effectiveness. Since biotechnology, which is gene knockdown, is being practiced, there is no need for buying chemicals or other hormones, which is used for sex reversal. Thus, there's a low production cost. Even though gene knockdown brings various positive impacts in aquaculture, however, it do bring some negative impacts to the industry. The first negative impact is the larger space requirement. In the context of Macrobrachium rosenbergii, these prawn species are territorial, especially the males. The males require more space to grow, thus, an all male production or a monosex production is a concerning issue due to its larger space requirement. However, if it's an all female production, it is an advantage because females can be stocked in high stocking densities, up to 4 times than the original stocking density if it was an all male production. Next is the off target effects. Sometimes in gene knockdown, there might be unintended gene silencing, where we silence unintended genes, which can affect the species physiology and behavior, which could affect the monosex population. The third negative impact of gene knockdown is RNAi resistance. There's a potential resistance to RNAi, which has a similar ideology as the antibiotic resistance. This may be ineffective in the long run. The fourth negative impact is the environmental impact, where there might be a spread to the environment. The presence of potential risk of dsRNA or double-stranded RNA spreading to non-targeted species in aquatic environment can disrupt the local fish genetic pool, which in the long run cause adverse effects to the fish species biodiversity or to the environment. To wrap up the presentation, gene knockdown is a very useful biotechnology method which can be applied in aquaculture. It brings various advantages such as monosex population, higher profits. At the same time, it does bring various kinds of negative effects, such as unintended gene silencing and environmental effects. Farmers or aquaculturists who would like to use this biotechnology method should understand the advantage and the disadvantage of this method and should use it wisely. So these are the lists of references used to prepare this presentation. So that's all from me, Doctor. Thank you so much for your time and years.